Hi, and welcome to Tom Kennedy Science, and I'm your host, Dr. Tom Kennedy. Today, we're going to talk about gene expression in prokaryotes. So what exactly is gene expression? Well, if you remember, your DNA stores information in the form of a gene. It's a sequence of nucleotides, right? All those A's, T's, G's, and C's. So if you have a sequence like G, A, T, T, A, C, A, that codes for specific information, in the case Gattaca, right? But at any rate, DNA doesn't make proteins. So inside of every cell, you transcribe your DNA to what is called a messenger RNA. We're basically just copying the sequence of DNA onto messenger RNA, and that is called transcription. Then, once your messenger RNA is made inside of a prokaryote, it will then bind to a ribosome. Now remember, ribosomes are the structures that make proteins inside of cells. And then we will translate the information in the messenger RNA into a protein. So we're going to take a sequence of nucleotides and we're going to translate that to a sequence of amino acids, right? So it's like translating a language, right? We take a sequence of words in English and we translate that to Spanish and vice versa. So we have transcription and translation. This is gene expression, okay? And in prokaryotes, it can be very quick. Now, your typical bacteria cell might have between 4,500 and 6,500 different genes to make proteins. And they're not going to express all of those proteins at once. So they use different types of proteins to regulate when they make those different proteins. Now, why would they regulate gene expression? Well, let's think about the bacterium in your gut. You have trillions, literally trillions of bacteria in your gut and about 1,000 different types. Now, they are totally dependent on what you eat. So you can imagine you drink a glass of milk. Well, then all of a sudden they have the sugar lactose available to them. You drink a regular soda like a Mountain Dew or a Coke. They all of a sudden have lots of fructose and glucose available to them. You go eat a yummy pizza for lunch. Now you've got fat, protein, and carbohydrates. Or let's say you want to be healthy and you eat a salad with a balsamic vinaigrette and some nuts on there. Well, every time you're eating a different meal, those bacteria, they require different enzymes to break down the different types of molecules you're giving them. And the point is they don't really want to produce all of their proteins at once. So one way that prokaryotes regulate this is called an operon. Now I'm going to come back and explain this diagram more, but an operon allows a bacteria cell, a prokaryote, to turn off and on gene expression for particular genes depending on what's happening to their environment. So you can think about it this way. You drink a sugary drink or milk. That's one source of energy. You go eat some salad. That's, a whole bunch of molecules right there, including cellulose and all these other molecules. The idea here is when you're switching your diet back and forth, these bacteria have to respond to what you're eating, right? So by using an operon, they can quickly respond to the environment by changing the expression of their genes and producing different proteins depending on what you eat. So they can rapidly respond to a changing environment. That would be your gut microbiome. Now, here's one thing that's important about gene expression. We often think of it as an on and off switch. And in fact, I'm even going to teach a Lac Operon like it's an on and off switch. That's not entirely the case. Gene expression is more like a volume knob. You can turn it almost completely off where you're not making any product, or you can ramp it all the way up to full blast and make the gene as fast as you can. And like I said, this depends on the needs of the environment. Now, wait, do you notice that knob? It goes to 11 because sometimes your amp needs to go to 11. And in fact, this came out of Spinal Tap, right? In Spinal Tap, Nigel Tufnell shows off his amp that goes to 11. We need that little extra push. Spinal Tap was an amazing spoof on metal music back in the 80s. And interestingly, Nigel Tufnell right there, his amp going to 11. He was a six-fingered, oh wait, how do I do? Yeah, he was a six-fingered man in The Princess Bride. That's pretty cool, isn't it? So The point that I'm trying to make is that gene expression, even though that we're showing it as an on and off switch, that's not entirely correct. It's more like a volume knob where you can ramp it up and down. So let's take a closer look at gene expression in a prokaryote. We're going to look at this operon. Now, first of all, there's not a lot of excess DNA in a prokaryotic cell. They're very, very streamlined, right? This allows for a rapid response to the environment, rapid reproduction, rapid evolution. So you've got a gene to make, let's say, the proteins to break down lactose. Now, in us, we call that lactase. So you also have these two other regions of your DNA, and they're right next to each other. You have what is called the promoter region and the operator region. So you got your gene right here. You've got in between that the operator, and then you've got the promoter. Now this is interesting. So what the promoter region does is it promotes transcription. So what we have is a promoter region. Promoter. What does it do? It promotes something. In this case, it's going to promote transcription. And what it does is, if you notice, there's something attached to it. That's your RNA polymerase. Poly means many. ACE is an enzyme. RNA polymerase is an enzyme that synthesizes RNA. Let me say that again. RNA polymerase is a protein enzyme that makes RNA. This is what transcribes our DNA to our messenger RNA. So in prokaryotes, the RNA polymerase easily attaches to the promoter region, and once it attaches to the promoter region, guess what? It immediately begins to transcribe the gene to make your proteins to break down lactose. Now, the point is, we don't always want to be making lactase if we don't have the sugar lactose. So if you're not drinking milk, the bacteria inside your gut, they don't need to be making any of the proteins to break it down. So how do you turn this off? Well, this is where the promoter region comes into play. Now, the promoter region is a sequence of DNA. So is the operator. Now, we're going to have a protein that's called a repressor. I wonder what a repressor does. Could it repress something like transcription? So if you think about it this way, I've got my RNA polymerase. It's binding to the promoter region. It's ready to transcribe. So the way we can prevent transcription, if you're not drinking milk, is to literally, physically block the RNA polymerase from transcribing. And this is what a repressor does. A repressor binds to the operator, okay? So if you're not drinking milk, you have what is called a repressor, and it's binding to that operator, and it's preventing transcription of that gene. Now this repressor, this is a protein. To be clear, promoter regions and operator regions, that's our control sequences. They are sequences of DNA, right? They're part of the bacterial DNA, whereas a repressor is a protein. And a repressor is a pretty cool protein, right? And remember, proteins often come in one of two shapes. It could be more, but in the case of a repressor, it can be active or inactive. And you make it active or inactive by changing its shape. So if I've got an active repressor, okay, it's binding to the operator. Now, if I want to inactivate the repressor by changing its shape, it'll come off the operator region and allow for transcription. So let's take a closer look. In this case, we've got a repressor. This repressor in a Lac Operon, for lactose operon, if you drink milk, you have lactose inside of you. And those bacteria will take up the lactose, and the lactose will bind to a specific area on the repressor and change its shape. And in this case, in case of a Lac Operon, lactose will inactivate the repressor. And by inactivating the repressor, it pops off the operator region, and then the RNA polymerase can begin transcription. And then it will start making these lactose utilizing genes, right, to break down lactose. So then as the cell uses up all the lactose, guess what? There's no more lactose to bind to the repressor. If there's no lactose binding to the repressor changes shape, binds to the operator region, and transcription stops again until you drink another glass of milk. Now, if you're like me, I'm lactose intolerant. Now that's lactose intolerant doesn't have anything to do with the bacteria in my gut, more with what I produce. Now me, I rarely drink milk. So if a bacteria actually has these genes inside of me, they rarely ever get turned on. Now, of course, there are different types of operons in bacteria, inducible and repressible operons. And I'm not going to get into the trip operon and how that's inducible here. That's for a more advanced class. But basically, I just wanted to cover the Lac Operon to show you how it works in a prokaryotic cell. To know that the main point here is a prokaryotic cell has all of these genes. They don't express them all at the same time because that would be wasteful. They'd spend all their energy making proteins they're not using so they can regulate it with an operon. And we always talk about the Lac Operon because, well, it was the first one discovered. And there's many more. Okay, until next time, we'll do gene expression in eukaryotes. And eukaryotes, they're bigger, larger, more complex. And guess what? Gene expression is way more complicated in eukaryotes than it is in prokaryotes. Makes sense. Until next time.